In Chapter 6, we overviewed just energy moving and different types of reactions. We talked a lot about enzymes. In Chapter 7, we are going to go in depth into the process known as cellular respiration. This involves many pieces, and this is mainly focused on how animals do respiration, particularly humans. But there are other ways of living. If you take microbiology, there are all different types of ways of producing energy. But we are going to look first at the energy in living systems. Then we will dive into the details of the different steps of the process of breaking down glucose. That includes glycolysis, then this thing called oxidation, pyruvate, and the citric acid cycle. Then we finally finish up with oxidative phosphorylation and how that produces large amounts of ATP. After that, we'll briefly look at some types of metabolism that involve no oxygen, anaerobic types of respiration, things like that. We'll talk briefly about how carbohydrates other than glucose, proteins, and lipids can feed into these pathways. And then we'll talk a little bit about the regulation of the cellular respiration processes. So, my favorite thing about ultra running, which my wife and I love to do, is that you get to go out and you're encouraged to eat as much as possible. It's great, right? You do a long run, you're burning 6,000 plus kilocalories, right? So you need a lot of nutrients to sustain you through that. The cell has a finely tuned machine to do this. We're going to talk about one piece of this, and this piece focuses on glucose. So this is our big overview here. This diagram is really key to understanding Chapter 7 and the respiration process. So the molecule glucose, right, has 6 carbons. It's got some hydrogen, oxygen. They're all bonded together. There's a kind of 3-step process to extracting energy from glucose. The first piece is called glycolysis. And this basically breaks glucose in half. A little bit of ATP gets made here. Pretty much every organism on Earth can do glycolysis. Of course, it's biology, there's exceptions, but pretty much everything can do glycolysis, and they do it the same way. Then we end up with 2 pyruvic acids here. These are 3 carbon sugars because we split it in half. This moves in animals into the mitochondria where some steps happen, and we start chopping carbons off of this molecule, producing CO2 in the process. We feed through something like the citric acid cycle, and again, a small amount of ATP is made. The real key here is that we are extracting high energy electrons at each of these stages, and that's going to feed into a process called the electron transport chain. These are going to pump hydrogen ions, building a gradient. We've talked a lot about gradients. We're going to build that gradient. And then we're going to allow them to flow through an enzyme called ATP synthase. ATP synthase is where we generate tons of ATP. So you'll see from one molecule of glucose, glycolysis made 2, the citric acid cycle made 2. This electron transport chain makes 32. So we are fully extracting the energy here. Before we get to that, though, let's talk energy overall. So I mentioned electrons there. We need to talk a bit about how electrons can transfer energy in living systems. And then we'll talk about how ATP gets used as an energy source. So remember, we mentioned it last time in Chapter 6 that adenosine triphosphate is a nucleotide, but it has these 3 phosphates on there, and the bonds in there have a really high energy potential, so we can store energy in there temporarily at least. So part of the energy bit of chemistry is the movement of electrons. We can think of electrons like energy. They can be transferred from one molecule to another. And when we transfer electrons, we call these processes redox reactions because they involve oxidation and reduction. I'll go on to define those here in just a sec. So we shorten it to redox because when you have one, you always have the other occurring. So what is this? Well, here we have a diagram. Substance A has an electron on it, and it is going to donate that electron to substance B. So A is going to lose an electron and B is going to gain an electron. So we would call this process of A losing an electron, we would call that oxidation. Loss of electrons is oxidation. So A is oxidized here. B, on the other hand, went from not having an electron to gaining an electron. That is reduction. Okay, substance B gains an electron, it has become reduced. Okay, a good way to memorize this is the mnemonic Leo says grr, or you can do this one for people of a certain age. This is Leonardo DiCaprio. You might not know him. Leo says grr, right? So loss of electrons, oxidation, gain of electrons, reduction. So in oxidation, whatever is donating an electron, losing an electron, and in reduction, it is accepting an electron, gaining an electron. Okay, so where does this happen? Well, in the reactions we're going to talk about, there are a few key molecules that we call high-energy electron carriers. These carry little small packets of energy, basically, that can be moved around incrementally. That part's important. Remember we talked about the marshmallow in Chapter 6 and how the marshmallow can light on fire, right? If you leave it in the fire too long, it bursts into flames because there's lots of energy stored in that glucose. But how does our body break it down in small chunks so that we don't ignite and burst into flames? Well, this moving small packets of energy around is how we get around that. We take little bits at a time, little bits, and that leads to a controlled release of the energy. One of these high energy electron carriers is this molecule called nicotinamide adenine dinucleotide, or NAD. Okay, NAD is technically positively charged. It can accept electrons, and in the process, some hydrogens come with it. Don't worry about that. But it becomes called NADH once it's reduced. So NAD plus is missing the electron. When it gains electrons, it becomes reduced, gain of electrons reduction, right? And we have NADH. NADH can donate those electrons to something else, and it will go back to being NAD. So we cycle between this. We have NAD plus, and it gains electrons, giving it a higher potential energy, and now we call it NADH there. And then that can donate electrons, going back to be NAD plus. There are some other electron carriers that get used in different processes. They do the same thing. They just have different names. They're different molecules. Again, why have multiple? Because this system evolved, and so you have to memorize multiple things because there are multiple things. So, FAD and FADH2, okay? This is the form without the electrons. We add the electrons, and we get FADH2, okay? This is related to NAD. And then there's an important one for anabolic reactions and photosynthesis. NADP and NADPH are involved in there. Again, this is the form without the electrons. We donate electrons to it. It becomes NADPH. Okay, so these are carrying electron energy in little chunks. How does this fit in with our other molecule? Well, ATP is a little bit longer term storage. So those FAD, NAD, NADPH, those are real short-term stores of energy. ATP is a little bit more long-term in context, right? Not as long as like a sugar or something like that, but it stores energy longer. So we have ATP, right? And we said that we can break the bonds, which releases energy, which can power endergonic reactions. And then to reform that ATP, we need energy from exergonic reactions to come in to rebuild it. So our big question in this chapter is, where is this energy coming from, right? I've been saying it comes from somewhere. Where does it actually come from is the key question. Well, this is the answer, okay? Job done. It's over, right? We're going to learn the pieces of this and how they contribute energy to build molecules of ATP in the process. All right, so with our newfound knowledge, right, we know that NADH and FADH2 are carrying electrons. That's filling in one piece of the puzzle here. Okay, we'll come back to this diagram many times, don't worry. So one of the things here, we said making ATP. Well, if we start with ADP, that has two phosphates, we need to put a phosphate back on. This is where a process called phosphorylation comes in. Phosphorylation is adding a phosphate to something. Simple as that. There's lots of different types, though. We're going to talk about two types where we can build ATP in the breakdown of glucose. Just to teach you more terminology, right? Dephosphorylation means removing a phosphate, right? So phosphorylation is adding, dephosphorylation is removing. I swear language makes sense, okay? We just add D to it and it's the reverse. So we're going to look at two types of phosphorylation here. One is called substrate level phosphorylation. That's where we directly just stick a phosphate onto ADP from an intermediate substrate. So this substrate has a phosphate on it, we carry it in, and it donates that phosphate to ADP, forming ATP. Remember, ADP is adenosine diphosphate. It's got two. Once we add one more, we get ATP. Okay. There is another way that ATP can get made, and that is through oxidative phosphorylation. Again, we will come back to this, but what is this? Well, in the mitochondria, there's all this space and surface area, and in the inner membrane here, okay, the inner membrane, we have the electron transport chain. High energy electrons get brought in. That powers pumps that pump hydrogen ions outside in between the two membranes here. Then those hydrogen ions are allowed to flow through an enzyme called ATP synthase. The ATP synthase turns and it catalyzes the reaction of adding phosphate to ADP. This is called oxidative phosphorylation because oxygen is involved in pulling these electrons through the electron transport chain. You'll notice there's no substrate bringing the phosphate in. It's just free phosphate that's floating around. So this distinction is not super critical, but I want you to see the two different places that this is happening. By far the most efficient is the oxidative phosphorylation, which is doing most of the work here. This process, we'll talk about it, is called chemiosmosis because we have the flow of hydrogen ions. It's kind of like osmosis for water. They use that term. It's ATP production from a proton gradient. We'll come back to that when we talk about this step in the process. All right, we need to review. If you lose a high energy electron from the molecule NADH, this would mean it is, go ahead and pause the video, think about these things. So, loss of electrons, oxidation would be our answer. Leo, loss of electrons, oxidation. Okay, it would maybe be sad, but it can gain another electron. Don't fear for NAD plus that is formed. Cellular respiration is going to involve several biochemical pathways. Each of those is going to remove a small amount of energy from glucose. We're going to do that in small little chunks so we don't explode or light on fire, basically. This is going to involve redox reactions, oxidation reduction reactions, which are going to involve transfer of electrons from one substance to another. Loss of electrons is oxidation. Gain of electrons is reduction. We can think of these electrons as transferring energy. They have energy. We talked about some high energy electron carriers. We will see those in the process. They are NADH, FADH2, and NADPH. These all have the electrons on them. Remember, they're co-conspirators, I guess. NAD+, FAD+, and NADP+, all are lacking electrons and can accept them, and they cycle between those two states. ATP is going to store energy in its phosphate bond. So this is a little bit longer term storage in the cell. To do that, to make ATP, we're going to need to have phosphorylation occur. That's addition of phosphate to a substance. We have substrate level phosphorylation. An enzyme directly takes a phosphate from one thing and adds it to ADP to form ATP. Oxidative phosphorylation, on the other hand, ATP is produced using energy from a hydrogen ion gradient, and it flows through ATP synthase, which will add inorganic phosphate just to ADP to form ATP. Okay, that is it for 7.1.